Dr. Doris Gabriel, R&D Manager at Apidel, Geneva, will talk about ApidSOL Micellar Nanocarriers for enhanced local drug delivery.
Okay, dear ladies and gentlemen, it's my pleasure to be here today and to talk about ApidSOL nanocarriers for local drug delivery. So ApidSOL nanocarriers are based on pegylated hexyl-substituted polylactic acid with a molecular weight of approximately 5.5 kilodalton. This polymer was originally invented at the University of Geneva here in Switzerland, and it's now developed by Apidel, a University of Geneva spin-off company. Now, when you look at this polymer, you can see it's an amphiphilic molecule, and in an aqueous environment, it forms micellar structures. And these micelles can encapsulate poorly soluble drugs inside them. So when we look at the drug product, these are transparent colloidal solutions. ApidSOL is a versatile delivery system for poorly soluble drugs, and it has some unique features. So among those, we have a small particle size with between 20 and 50 nanometers. We have an inert PEG surface, which minimizes interactions with the extracellular matrix. The hex PLA part of the polymer is degradable and results in the release of safe fragments. We have good tox profile. These particles were tested in several species using multiple administration routes. Then with respect to applicability, it can be used to formulate a wide range of hydrophobic drugs. It efficiently solubilizes them, and we have enhancement of apparent solubility of up to several thousand times. And the manufacturing process is basically just a self-assembly process. It's upscalable, and we have also a poster outside which describes the upscale from milliliter scale to liter scale. Now, this slide to depict how ApidSOL nanocarriers improve bioavailability and therapeutic profile. So first of all, the drug is solubilized inside the nanocarriers. And then these nanocarriers facilitate transport of the drug into biological structures, such as the skin or mucosal surfaces. In this biological environment, we have dilution of the micelles, and eventually they can release the drug and form a local drug depot. So by doing so, we can achieve high local drug levels, and this can actually improve therapeutic efficacy if local drug levels are an issue, or it can be used to reduce the dosing frequency, or we can also use this to reduce the dose in the drug product. Now, Apidel's focus is on local drug delivery, and we have several products in our development pipeline with applications in ophthalmology, dermatology, and also gastroenterology. Today, I would like to focus on gastroenterology and more specifically on inflammatory bowel disease. Now, there are roughly 1 to 2 million cases of inflammatory bowel diseases in the US, and more than 50% of these patients have ulcerative colitis. The remaining patients suffer from Crohn's disease. Now, ulcerative colitis is restricted to the large intestine, and staging is according to inflammation severity and extent. Physiology is altered, so we have mucus hypersecretion, and also the epithelial permeability is increased. There is a role for local treatment in this disease because approximately 78% of patients suffer from distal colitis, which can be treated locally by application of enemas. Now, current pharmacological treatments include local and oral anti-inflammatory drugs, systemic biologics, and also oral and systemic immunosuppressants. Depending on the disease severity, we have combinations of these treatment options. Now, when we look at cyclosporin A, this is actually a potent immunosuppressant. It has demonstrated therapeutic efficacy in even refractory cases of ulcerative colitis. On the downside, we have severe systemic side effects, and local application is actually difficult because we have low solubility of this molecule and subsequently low tissue-specific bioavailability. So we thought, okay, this could be interesting to develop cyclosporin ApidSOL enema formulations with the rationale to deliver high local doses into these inflamed colon tissues and to reduce systemic side effects by limiting exposure, systemic exposure. So we formulated cyclosporin ApidSOL formulations of different doses, so 0.01, 0.1, and 1%. And we tested these formulations in an animal model of ulcerative colitis in the TNBS-induced inflammation model. So animals received the different doses for 7 days, and at the end of this treatment, inflammation was staged. So we have a macroscopic score, histology, and also a biochemical marker, which is the myeloperoxidase. Now here you see the results for the macroscopic scoring. So actually, the higher the bars, the more inflammation we have. So we can say that we have the best therapeutic effect with the 0.1% cyclosporin formulation. Actually, on the histology level and with respect to the biochemical marker, we have also the same findings. So the 0.1% formulation performed best. Now, in the next step, we were interested to assess how much do we deliver to the colon tissues and how much do we have in systemic circulation. And we measured drug tissue levels by UPLC-MS, and you see here the data for the 0.1 and the 1% formulation. So we have healthy animals and healthy control animals and the animals with the ulcerative colitis. And what you can see is that actually with both those doses, we have much higher levels in the colitis animals. We can also see that with the higher dose, we can deliver much higher tissue levels into these inflamed tissues. Importantly, we did not observe any systemic exposure, so we had no detectable drug plasma levels for the animals treated with 0.1% formulation. We investigated then in the next step colon tissue distribution and penetration depth using fluorescent micelles. So we loaded these micelles with a red fluorescent dye and treated animals with these micelles. You see now here on the left tissue sections of animals, of healthy animals, and on the right animals which suffered from ulcerative colitis. The blue dots are actually DAPI-stained nuclei. What we can observe is that we have much less red fluorescence in the healthy animals and more fluorescence in the ulcerative colitis animals, which is actually consistent also with the quantitative drug dosing data. With respect to tissue distribution in the inflamed animals, we have red fluorescence in the remainders of the mucosa here. Then we have also fluorescence in the submucosa and even in the muscularis. Of course, also toxicity is an important issue. So we tested this formulation in vitro and also in vivo using different administration routes. So we have tested them in gastroenterology, so these topical enema formulations. Then we have also repeated administrations in ophthalmology and also dermatology, and we found even after repeated dose dosings, we found no signs of local or systemic toxicity. Okay, and this brings me already to the summary. So I hope I could show you that these ApidSOL nanocarriers are biodegradable and safe. They efficiently solubilize drugs. They deliver them into tissues after a local application and enable the formation of a local tissue depot. They can achieve high local doses and can, by doing so, reduce the drug dosing frequency or the drug product dose, and they minimize drug exposure and side effects. So thank you for your attention, and I'm happy to answer questions you might have.
Thank you very much. First question, please.
Hi, and thank you for the nice work presented. I have a question regarding the mechanistic issues behind the use of this. I guess we are talking about micelles. So on the one hand, you are claiming that using this kind of micelles, you are forming a depot onto the skin or the eye, and you are facilitating the transport of the associated drug. But at the same time, with the oral application, with the mucosal application, you are claiming that you form a depot and there is not absorption. So that sounds to me like antagonistic performances depending on the modality of administration. So
actually the gastrointestinal application was a rectal application. It was a topical application.
Yeah, but in that case, you are not claiming and improved transport of the drug. You are claiming that you are localizing the drug there and there is no systemic absorption, whereas in the case of the skin, you are claiming that using this nanocarrier, you are facilitating the transport.
Okay. Yeah.
So in one case,
it's actually a slightly different situation because we have a leaky barrier in this case. But what we do in this case is mostly solubilizing the drug and making it available to go into this tissue. But I would agree with you that the transport, the facilitated transport is maybe a little bit less important in this specific application.
So why the cyclosporin A, when you administer it intrarectally, doesn't go to the blood circulation as it does the marketed formulation? Why?
Why it does not go? Because I think it's released from the micelles. Once it's inside the biological structure, it's not soluble anymore. That's the only explanation I can give at this stage. But we didn't measure any plasma drug levels.
I was going to ask if you've seen any anti-inflammatory properties really from, particularly I guess in the enema cases, from the carrier itself, you know, the PEG action, which is in the literature. And I've actually personally seen it in assays as well.
In this, I didn't. Well, I think I showed the data also for the carrier, so we didn't observe anything just for the vehicle itself. At least no significant difference.